Thank you for the introduction. I'd like to express my gratitude towards, oops, sorry. I'd like to express my gratitude towards Rick for giving me an opportunity to present here today. The topic of my presentation today is the structure-function relationship of human AT1 R transferase enzyme, which was published in Nature Communications in July of this year as a collaboration between Case Western Reserve University in the United States and Seoul National University in Korea. As a co-corresponding author, I was responsible for the cell biology experiments on this paper. And because I am a cellular and molecular biologist by training, please bear with me if my explanation of the structural biology aspects of this paper are a bit lacking. My research focus is on cellular proteolysis and degradation as a means of not only constitutive upkeep, but also as a cellular defense mechanism. Specifically, I am interested in degradation determinant systems that modulate cellular proteolysis. The term degron, meaning degradation signals, was coined by Dr. Alexander Varshavsky more than 25 years ago. He founded and pioneered the field of endogram pathway, in which specific amino acid residues and their post-translational modification results in protein degradation. Recently, the C-degram pathway was also discovered. Currently, there are 5 endegron subpathways, the formyl methionine, glycine, acetyl, proline, and arginine branches. In each of these branch subpathways, there is a specific clientele or a set of amino acid residues and their post-translational modifications that dictates the in vivo stability of substrate proteins based on the identity of its N-terminal amino acid residue. Our lab, under Professor Yong-tae Kwan of Seoul National University, has been studying the mammalian arginine endogram pathway for more than 25 years. Within this pathway, the N-terminal amino acid residue exposed after N-terminal methionine cleavage is considered as type 1 or type 2 if they are positively charged and basic or bulky and hydrophobic. Type 1 endegrons in this case are arginine, lysine, and histidine, while type 2 endegrons include phenylalanine, tryptophan, tyrosine, leucine, and isoleucine. In addition to the arginine residue that is naturally found after N-terminal methionine cleavage or at the beginning of C-terminal fragments, specific amino acid residues can be post-translationally conjugated with N-terminal arginine. Specifically, N-terminal asparagine and glutamine can be deamidated via NTAN1 and NTAQ1, respectively, to yield aspartic acid and glutamic acid. These residues are then selectively recognized by the AT1 R transferase enzyme, which conjugates L-arginine from high-energy arginal tRNA molecules to the arginylation-permissive N-termini of substrate proteins. Additionally, the N-terminal cysteine residue can be oxidized by oxidases as well as reactive oxygen species into sulfinic and sulfonic acid, which are then N-terminally arginylated by AT1. Thus, depending on their status, endegrons can be characterized as tertiary, secondary, or primary. Due to the scientific community's interest in the ubiquitin proteasome system in the late 1990s and the early 2000s, the first endogenous substrates of the mammalian arginine endogram pathway were tested for proteasomal degradation. A quick and fun, I guess, side story is that the 2004 Nobel Prize in Chemistry, which was awarded for the discovery of the ubiquitin proteasome system, could have been, and almost was awarded to Dr. Varshavsky, because some of the first confirmed substrates of the ubiquitin proteasome system were from the endogram pathway. So anyways, once type 1 and type 2 endegrons are exposed, they are recognized by specific receptors called N-recognins. The very first N-recognins discovered in the arginine endocrine pathway were found to contain the UBR box and N domains, which respectively and selectively recognize and interact with type 1 and type 2 endocrines. Additionally, these so-called UBR recognins were found to be also canonical and non-canonical E3 ligases containing the ring domain for UBR1, 2, and 3, or the hex domain for UBR5. Thus, these UBR recognins / E3 ligases simultaneously recognize endegrons via the UBR box or N domain, and also subsequently simultaneously ubiquitinate them for proteasomal degradation. In this sense, the N-terminal arginine is a proteasomal endegron. Our lab in 2015 also discovered that the N-terminal arginine residue can modulate the lysosomal proteolysis of cognate substrates via the autophagy lysosome system, specifically macroautophagy. We found that certain endoplasmic reticulum chaperones, such as GRP78 or BIP, could be retrotranslocated from the endoplasmic reticulum back to the cytosol after their ER signal peptide is cleaved, leaving them prone and open to N-terminal arginylation by ATE1. The resulting arginylated ER chaperones were targeted to and degraded by autophagy. And additionally, due to their pre-existing chaperone activities, these originally ER chaperones also accelerated the autophagic sequestration and lysosomal hydrolysis of cytosolic protein aggregates. During this process, we found that the archetypal autophagy cargo receptor P62, also known as sequestosome 1, functions as a novel autophagic N-recognin via its ZZ domain. The amino acid sequence of the P62 ZZ domain was evolutionarily conserved in mammals and showed close resemblance to the UBR box domain of UBR E3 ligases. Interestingly, and unique to P62, once the N-terminal arginine binds the Z domain of P62, P62 undergoes a conformational and biological activation, resulting in self-oligomerization into liquid-liquid phase separation, or LLPS, filaments. Thus, the P62 / N-terminal arginine complex, including the cognate substrates and their binding partners, are initially sequestered into temporary P62 bodies that are then targeted to maturing autophagic membranes for eventual acidic hydrolysis in the lysosome. In addition to accelerating the autophagic targeting and lysosomal hydrolysis of substrates and their binding partners, we found that the N-terminal arginine residue also facilitates induction of selective autophagy, specifically via autophagous biogenesis for selective autophagy. Until recently, from the late 2000s and onwards, bulk autophagy was initially investigated as a starvation-induced and non-selective process for nutrient and energy metabolism. However, selective autophagy is also vital as a cellular defense mechanism for wholesale degradation of unwanted, damaged, and/or pathological proteins, their aggregates, organelles, and even pathogens, among many other types of cargoes. We found that activated P62 translocates to the ER surface to the site of autophagous biogenesis, where it mediates the neogenesis of new phagophore membranes via novel mechanisms. Through both induction of autophagous biogenesis in selective autophagy and acceleration of P62 targeting, the arginine endegron pathway was shown to mediate the autophagic degradation of not only protein, but also other types of cargoes, majorly most well exemplified by organelles such as earphagy, lipophagy, xenophagy, or and mitophagy. So as you could see, the biomolecular and cellular biology aspects, phenomena, principles, and mechanisms of the arginine endogram pathway have been relatively well elucidated. I'd like to switch gears and now talk about the arginine endogram pathway from a structural biology standpoint. Currently, excluding the plant endogram system, key structural biology studies of the yeast and human of the yeast and human endogram pathway have focused on yeast UBR1 and yeast NTA1 from 2010 to 2016. Once the autophagic arginine endogram pathway in humans was discovered in 2015, the crystal structure of the P62ZZ domain that facilitates recognition of N-terminal arginine and other endegrons was independently resolved in 2018 by two groups, one of which is us, and the other Professor Song Hyun-kyu of Korea University. Most recently in 2022, Professor Song's group in Korea University was able to elucidate the structure of yeast AT1. However, to date, the three-dimensional structure of human and mammalian AT1 had not been resolved and was only analyzed by molecular modeling. Thus, we wanted to resolve the actual three-dimensional structure of human AT1 and compare it to that of yeast AT1. Yeast AT1 adopts a compact fold and primarily interacts with the major groove of the tRNA acceptor arm via its conserved GNAT, GNAT domain. The N-terminal and C-terminal, C-termini of yeast AT1 converge to form a putative substrate binding domain, featuring a zinc finger binding motif that is conserved in Cluiveromyces lactis. In contrast, human AT1 only shares 32% sequence identity with yeast AT1, and the two enzymes share only a few common protein substrates, which results in a significant gap in our understanding of the catalytic function, substrate selection, or the molecular interactions that regulate AT1. And conserved from yeast to plant and mammals, the AT1 is an enzyme that covalently transfers an arginine from tRNA molecule to its protein substrate and has been implicated in a multitude of biological processes, including prominently protein degradation. But unlike the other endogram systems, the arginine endogram branch is unique as if the action requires AT1 mediated protein arginylation in a tRNA dependent manner. Specifically, AT1 transfers the nutrient arginine from high energy arginal tRNA molecules to substrate proteins containing an exposed N-terminus. The arginal tRNA synthetase, arginine TRS, then recharges the arginal tRNA released from the reaction. Therefore, this mechanism of N-terminal arginylation inherently links fundamental cellular processes, including protein synthesis to nutrient availability and metabolism, and finally to protein turnover and degradation. Mammalian AT1 isoforms undergo alternative splicing of exons 1, 2, 8, and 9 to produce 4 different isoforms in humans. All 4 isoforms are capable of N-terminal arginylation and share critical residues or structural characteristics such as active site locations. However, for the purpose of this study, we focused on the isoform 2 variant of AT1 after screening for its in vitro arginylation capabilities. We first conducted LCMS analysis using a reconstituted arginylation reaction mixture or with purified recombinant AT1. And similar to previous findings with yeast AT1, the incubation of human AT1 alongside in vitro transcribed, human arginal tRNA and the model peptide substrate arginine RS, which was derived from the actin sequence, and finally with ATP, led to the detection of the anticipated arginylated product, with 156 Dalton mass increase. And interestingly, but consistent with previous mouse AT1 studies, intact protein analysis of purified AT1 revealed a secondary but minor peak that corresponds to a monoarginylated AT1. As the recombinant AT1 that we used in this study was produced as a GST fusion protein without an exposed N-terminus, this finding suggests that arginylation occurred at an internal residue of the peptide substrate. Considering the challenges in enriching the small arginylated fraction of AT1 for further analysis, we created an in-bacteria arginylation assay. Briefly, a 6-histidine tagged protein of interest model substrate was fused to the plant sumo and co-expressed with human AT1 and the ULP1 protease in E. coli. And following protease cleavage, the protein of interest exposes a selectable N-terminal residue that is amenable to arginylation. And this design facilitates targeted arginylation to enrich for modified product. So, and when we, perform this assay, the predominant peak of the purified model substrate sample aligned with the anticipated monoarginylated form. However, both the unmodified form and a double arginylated variant were identified. So to pinpoint whether arginylation then occurs internally or at the N-terminus, we performed 1H and 15N heteronuclear single quantum coherence, HSQC, spectral analysis. The HSQC spectrum of the unarginylated model substrate, when expressed with AT1, aligned with its N-terminally arginylated counterpart, which supports the notion that human AT1 primarily arginylates the N-terminal amino acid of its substrate, whereas the mid-chain, side-chain arginylated products are in lesser quantity and undetectable, basically undetectable. We then elucidated the structure of human AT1 to better understand its structure-function relationship, and also to understand the evolutionary divergence from East AT1. Using cryo-EM, we achieved an average resolution of 2.8 angstroms for the full-length apostate structure of human AT1 in dimeric form. Similar to East AT1, the N and C termini of human AT1 converge to create the substrate binding domain, which lies adjacent to the GNAT domain. Human AT1 also contains the conserved zinc finger motif within the substrate binding domain. And importantly, in stark contrast to East AT1 and other similar related amino acid dependent tRNA transferases, human AT1 forms a near perfect symmetric homodimer with an extensive surface of approximately 1,200 angstroms. Additionally, as compared with East AT1, human AT1 lacked the variable domain, but instead included an additional unstructured loop between residues 105 to 268, residing between helix alpha C and beta 6 in the GNAT domain. To better understand what this unstructured loop was, we superimposed our cryo-EM structure of human AT1 onto the AlphaFold computational model of human AT1. The extended loop is predicted to contain 3 long and 3 short alpha helices, which would be expected to interact with both the substrate binding and GNAT domains of human AT1. Further sequence analysis of the conserved helix in this loop, the amino acids 191 to 220, identified a notable presence of positively charged lysine and arginine residues. And interestingly, this positively charged region was shown to cluster to the same side of helix alpha C on the GNAT domain, which is a conserved tRNA binding element in East AT1 and other similar tRNA-dependent amino acid transferases. So we thus suspected that this alpha C loop helix facilitates the tRNA recognition in human AT1, and so conducted EMSA gel shift assays to determine the binding of wild type or mutant AT1 lacking this alpha C loop to the acceptor arm alone or the A and T arms of tRNA. And similar to East AT1, wild type AT1 displayed stronger affinity to A and T arms rather than the acceptor arm alone. And importantly, the mutant AT1 lacking the alpha C loop helix showed a 2 to 4 fold decrease in binding affinity to the A arm alone or the A and T arms, suggesting that the alpha C loop present in only human AT1 does indeed directly affect tRNA binding. Next, we reconstituted the AT1 tRNA substrate tripartite complex using human AT1, the in vitro transcribed human arginal tRNA, and the model arginine R substrate. Structural characterization by cryo-EM was achieved with an average resolution of 5.7 angstroms for the tripartite complex, and quantification of computationally selected particles, randomly selected particles, indicated that the inclusion of the tRNA and the substrate results in a substantial decrease of AT1 of the AT1 dimer population, dropping from 74% in the aqueous state to 21% in the presence of tRNA and substrate peptide. This finding suggests that the AT1 dimer dissociates upon binding of tRNA and or the substrate. In addition to the alpha C helix that interacts with the acceptor arm of tRNA, which is a common feature shared with East AT1, human AT1 also uniquely established a secondary contact site with the D and T arms of tRNA that stabilizes the divergent long loop containing the alpha C helix loop. Additionally, substrate engagement seems to induce an outward repositioning of the zinc finger, likely facilitating the accommodation of substrate to the correct position in the active site. To evaluate the contribution of individual sites to tRNA binding, we engineered a K88E and K92E variant mutant featuring charge reversal in the alpha C helix, which is supposed to interface with the major groove of the tRNA acceptor arm. So the K88E and K92E mutant showed approximately a twofold reduction in binding affinity to the A and T arms as compared to the alpha C loop variant. This observation supports the idea that interactions with the major groove of the acceptor arm of the tRNA are crucial for its binding to AT1. And this aligns with the conserved sequence of the alpha C helix in East and other tRNA-dependent amino acid transferases. And so to determine whether human AT1 specifically targets substrates in the endogram pathway dependent on this alpha C loop helix, we measure the proteasomal flux of a known endogram pathway substrate known as RGS4. In wild type and exon 5 deleted AT1 knockout HeLa cells, CRISPR-Cas9 generated HeLa cells, following proteasome inhibition via MG132 treatment. And as you can see, AT1 knockout cells exhibited no RGS4 accumulation post MG132 treatment, whereas wild type cells showed significant accumulation with a normalized PF index of approximately 29, and transient re-expression of wild type AT1 was able to restore this proteasomal flux index, while the RNA binding deficient mutant could not. And in line with this observation, immunostaining of recombinant tagged RGS4 indicated elevated levels in AT1 knockout cells that were rescued only by wild type, but not the mutant AT1. These results affirm that the electrostatic interactions between the alpha C helix of the unstructured loop and the bound arginal tRNA molecule are essential for human AT1 function. And since the resolution of the cryo-EM map of the ternary complex does not allow for side chain positioning, we then integrated computational modeling to improve our prediction of the molecular interactions that are critical to AT1 function. All-atom molecular dynamics flexible fitting, MDFF, using an explicit solvent model, was applied to MD simulations for an additional 200 nanoseconds. And in doing so, the structural model predicted that the A76 amino acid of the arginal tRNA is situated close to the arginine binding pocket at the opposite side of E387 in human AT1. And the space between A76 and E387 from end to end is approximately 13 angstroms, which is more than sufficient to accommodate the N-terminal arginine residue. To accommodate the arginine residue existing in the arginal tRNA molecule. And AT1 residues surrounding the pocket around the first two residues in the substrate peptide displayed a volume increase of 258 angstroms, which aligns with the observed outward shift of the zinc finger. Additionally, microscale thermophoresis assays demonstrated that in the absence of RNA, tRNA, the binding affinity of human AT1 for the arginylated peptide product is greater than for its unmodified peptide substrate. And the reintroduction of tRNA greatly enhances both the binding affinity of both the product, the arginylated product, and the substrate, unarginylated substrate peptide by respectively about 15 and 5-fold. And so to further interrogate the functional roles of the identified substrate binding sites, we carried out targeted mutagenesis of key residues. These mutations also led to a stabilization of RGF4 levels, corroborated by immunostaining. Additional mutations at E387A and K419A, as well as R56E, yielded similar outcomes. And consistent with our previous finding that the zinc finger domain is important for East AT1 stability and substrate binding, mutating the zinc coordinating residue C26 of human AT1 to alanine also led to RGS4 stabilization. These results emphasize the importance of the identified binding pocket for the recognition of N-terminal arginine, as well as the substrate N-terminal glutamic acid and aspartic acid. In human AT1, dimer formation is a unique attribute compared to AT1 from yeast or in related tRNA-dependent amino acid transferases. Our analysis of the dimeric interface identified an array of interacting residues important for substrate binding and catalysis. The dissociation constant measured by microscale thermophoresis for dimerization was found to be 8.5 micromolars in vitro. And to evaluate the functional significance of this dimerization property, we selected two sets of mutations to disrupt the dimeric interface of AT1. The first was informed by sequence alignment with East AT1, and the second set was selected for their likely probable electrostatic interactions with E308 and E309 of the opposite protomorph. We hypothesized that these mutations would expose large surface areas of AT1 to the solvent, which would then likely compromise protein stability. And in line with this hypothesis, this prediction, both pairs of mutations led to reduced melting temperatures to approximately 40 degrees Celsius, while the quadruple mutant also displayed a similar decrease in melting temperature. And native gel electrophoresis using AT1 knockout CRISPR-Cas9 HeLa cells corroborated this finding, showing that the quadruple mutant failed to form dimers, which is in direct stark contrast to the wild type variant. And to further explore the functional impact of mutations at the dimeric interface of AT1, we examined the proteasomal flux of RGS4 in AT1 knockout HeLa cells carrying these various mutations. Partial inhibition of RGS4 degradation was evident in cells expressing the W38F and H40E mutant, double mutant. But in contrast, the K476E and the R477E, as well as the quadruple mutant, not shown here, showed significant inhibition of RGS4 proteasomal flux. And immunocytochemical analysis of recombinant RGS4 also corroborated these findings. We thus reasoned that the AT1 dimerization might be a pivotal step in maintaining protein stability prior to substrate recognition and interaction in the arginine endogram pathway. And in support of this notion, co-immunoprecipitation assays highlighted a substantial difference in RGS4 interaction to AT1 in the triple mutant as compared to its wild type counterpart. And to further validate these observations, we then performed a proximity ligation assay, PLA assay, using recombinant RGS4 and the various human AT1 constructs. The PLA assay, the PLA technique, enables the detection of protein to protein interactions that occur within approximately 40 nanometers of each other. And consistent with prior data, mutations at the dimeric interface disrupted RGS4 AT1 complex formation. And these data collectively suggests that the dimeric interface of human AT1 is essential for its catalytic activity, presumably by stabilizing the AT1 protein prior to its substrate binding. So, to summarize, in this study, we unveil the 3D structures of human AT1 with and without a tRNA cofactor and a peptidic substrate. And importantly, human AT1 forms a symmetric homodimer in its apostate. And compared with its yeast counterpart, the human AT1 lacks the variable domain, which packs onto the substrate binding domain, which likely contributes to the increased stability of, in the case for yeast AT1. In this sense, the homodimerization property of human AT1 may be a means of stabilizing the stabilizing AT1 prior to its substrate binding. And another distinct feature of human AT1 is its extensive interactions with the original tRNA molecule that extend beyond the interactions that were previously observed with yeast AT1. I would like to acknowledge Professors Yi Zhang and Derek Taylor from Case Western Reserve University, as well as Professor Yong-Taek Han at Seoul National University for this collaboration. Thank you for listening.